and the organization page. So we'll have that in this. Okay, so let's begin. Good morning, everyone, and welcome to Bioinformatics for Precision Medicine. This program will highlight biomedical applications of big data bioinformatics in the context of major diseases like cancer and chronic infection. For those joining us for the first time, my name is Ilya Brodsky. I am the co-founder and CEO of Pine Biotech. Our vision is to enhance human health and well-being by enabling biological research and discovery with relevant data, solutions, and support. Our mission is to simplify bioinformatics and advance research through our modular and intuitive multi-omics analysis platform powered by human experience and artificial intelligence. We offer training, research support, and big data analytics tools designed for research and biomedical discovery. Our mission is to continue to support cutting-edge research and help solve big data challenges by developing innovative solutions that are easy to use and provide training to use them effectively and arrive at sound and reproducible results. I am honored to have been working at Pine Biotech with many others. Many of our team members have much more experience in this area than I, including Pine Biotech co-founders, Dr. Alfred Tauber, who comes from a medical background with expertise in oncology, immunology, and biochemistry of inflammation. He has a long-standing recognition for his generous support of many international projects and research activities. One of these projects is the establishment of the Tauber Bioinformatics Research Center at University of Haifa. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. He has numerous publications on bioinformatics and over the course of his career has developed multiple novel algorithmic approaches for biomedical data analysis. These have been widely used in basic and translational research in biotechnology, pharmaceutical R&D, agriculture, and biomedical research. What we will discuss today is also the result of hard work by some of the people mentioned here on the left. Julia Panov, who is an expert bioinformatician and has helped prepare many of the materials that we will use in this course. Avi Titievsky, who is a computer scientist and leads the development of the T-Bioinfo platform that we will utilize. Mohit Mazumdar, who is in charge of business development for Pine Biotech internationally. Joshua McKendall, who maintains the educational web portal we will leverage, and Bipsha Biswas, our online community manager and support specialist, who I am sure many of you had already had the chance to interact with. The program is designed to highlight the role of big data, bio and chemoinformatics. These are shaping modern biomedical innovation where molecular data is used for precision diagnostics. Big data integration and machine learning are transforming personalization of treatment selection and innovative algorithms that allow for unprecedented capabilities with drug discovery, design of large molecules, and drug repurposing and repositioning. Molecular data is routinely used in medicine for patient stratification and diagnosis of diseases like breast cancer. Because many patients have been diagnosed using these panels in long-term clinical trials, clinicians can assess the risk of cancer recurrence and survival based on information that they provide. In this chart, you can see a Meyer-Kaplan curve of survival and recurrence based on two clinical gene panels for breast cancer, PAM50 and Oncotype DX. These panels separate patients into three groups: high, medium, and low risk. Based on these assessments, a treatment can be selected. Traditional treatments for cancer are surgery, radiotherapy, and chemotherapy. Each one of these treatments can have multiple harmful side effects and is known to be effective in only specific situations. For example, tumors are typically irradiated before surgery, or if they are considered to be somewhat benign. Chemotherapy targets cell proliferation and often affects many other cells in the body. So not just the tumor, causing severe discomfort to patients and in some cases leading to complicated and even recurrence or emergence of new cancers. However, since the 1990s, a new type of treatment started emerging in clinical use called targeted cancer therapy. One such therapy is called Herceptin or trastuzumab. This is a monoclonal antibody that blocks the HER2 receptor, preventing it from receiving the growth factor and slowing cell growth. Since then, immunotherapy has transformed cancer care. Many of such precision drugs are the result of biomedical research studies of the tumor and its microenvironment, mapping the changes that characterize tumors and the novel understanding of the biological pathways inside tumor cells, as well as signaling pathways between tumors and their microenvironment, have led to multiple innovative interventions, blocking, suppressing, and enabling existing mechanisms of action to mitigate the effects of uncontrolled proliferation and metastasis. Opdivo, also called nivolumab, is one such example. This drug was approved for medical use in the United States in 2014. It is on the World Health Organization's list of essential medicines, the safest and most effective medicines needed in a health system. Opdivo is a medication used to treat a number of types of cancer, including melanoma, lung cancer, renal cell carcinoma, Hodgkin lymphoma, head and neck cancer, colon cancer, and liver cancer. But these benefits require multiple biomarkers to guide treatment selection and evaluate their effectiveness, placing data analysis at the center of such precision treatments. For example, nivolumab is used as a first-line treatment for inoperable or metastatic melanoma in combination with another drug called ipilimumab, another monoclonal antibody, if the cancer does not have a mutation in the BRAF gene. It is also used as a second-line treatment for inoperable or metastatic melanoma following treatment of ipilimumab, and if the cancer has a BRAF mutation, a BRAF inhibitor. It is also used to treat metastatic squamous non-small cell lung cancer with progression in or after platinum-based drugs and for treatment of small cell lung cancer. It is also used as a second-line treatment for renal cell carcinoma after anti-angiogenic treatment has failed. So you can see how these treatments, bringing novel precision, really require constant use of biomarkers and very detailed understanding of molecular processes that happen at the subcellular level. That's why data, its analysis and integration, is at the center of modern precision medicine. Researchers, biologists, doctors, and clinical lab technicians are trained to understand and utilize complicated molecular, clinical, and other high-throughput research data to diagnose disease, as well as develop, use, and evaluate treatment efficacy. Growth in data generation, specifically when it comes to high-throughput sequencing of cancer genomes and transcriptomes, has produced a big data problem that precludes many cancer biologists and oncologists from gleaning knowledge from these data regarding the nature of malignant processes, as well as the understanding of the relationships between tumor genomic profiles and treatment response. In this program, we will focus on such data, learning about the various types of omics data, including data that describes intracellular and intercellular processes associated with disease. We will also look at the role of pathogens and microorganisms in our bodies and the environment we live in to understand risks they pose to cause acute infectious disease and chronic risks that destabilize the immune system and lead to cancer-associated factors. And we will look at data sets that allow us to study the relationship between clinical and phenotypic data and understand the molecular biology characterized by omics data. What is often underappreciated in programs designed around cancer is the increasing role of viruses that we now understand who pose a tremendous risk for cancer to develop and complicate treatment. The Epstein-Barr virus, formerly called human gamma herpes virus 4, is one of such viruses, one of the most common viruses in human. It is best known as the cause of infectious mononucleosis, or mono. It is also associated with various non-malignant, pre-malignant, and malignant Epstein-Barr virus-associated lymphomas. The virus is also associated with the childhood disorders of Alice in Wonderland syndrome and acute cerebral ataxia. And based on some evidence, higher risk of developing certain autoimmune diseases such as lupus, rheumatoid arthritis, and multiple sclerosis. About 200,000 cancer cases per year are thought to be attributable to EBV. Another prevalent DNA virus is human papillomavirus from the Papillomaviridae family. Approximately 5% of all new cancers diagnosed in 2002 were attributable to HPV. Another such pathogen is hepatitis B and C, the most common risk factor for liver cancer. About 60% of liver cancer diagnoses are due to causes of chronic hepatitis B, leading to over 700,000 deaths from liver cancer annually. So that's why this Bioinformatics for Precision Medicine is a program designed to introduce the topics of basic and translational oncology, to study next generation sequencing, understanding DNA variants and the mutations that we can detect in DNA. We will talk about next generation sequencing for transcriptomics, where we can analyze gene expression patterns and see the difference in gene expression between multiple conditions, and also appreciate the role of machine learning in analysis of RNA-seq data. We'll talk about different types of projects, looking at different resources and public databases. We'll talk about genomics in virology and how we can study viral genomes inside the human cells and in other hosts. We'll talk about the process of multiple sequence alignment and evolutionary studies for viruses. And we'll be able to go in depth and discuss different pipeline results, the annotation of those results, and biological interpretation. We'll also talk about some of the challenges that make infectious diseases such an increasing risk and how they are related to cancer. And finally, we'll talk about the basic biology of host-pathogen interaction that is important to understand as you look at these causes of major chronic diseases. But before we go on, I wanted to give you an overview of some of the resources that we've prepared in this program and guide you to the various links where you can see some of those and start interacting with the program resources. The primary location for all of these resources together is going to be on this educational portal, edu.t-bio.info. On this portal, you will see an organizational page where we have a home page that describes the process of the different sessions and how they are organized in the program. Gives you access to the forum where we will post recordings of these sessions and interact with you to discuss different topics and challenges that you're facing. You will have a list of courses that you can see here in the back that are going to be provided for all of the participants of the program. Some project examples that we will review, where we will utilize data sets from published journals. You will see progress and keep track of where you are related to your peers in the program, and see other elements of the program that will help you make the most out of this Bioinformatics for Precision Medicine program. So let's take a look. Once you have registered, you should have received an email on how to access this link. In this link, you will see a number of different resources. For example, right here, you will see the upcoming events. Today, we will talk about Introduction to Translational Oncology Research. You can also navigate to future events and see what's coming up. Down here, you will see activity from participants of the program. For example, Beepsha recently posted a topic, welcome to the forum, and session recordings. Those are going to be available right here on the forum tab. So on the forum tab, you will see recordings of the sessions, you'll see highlighted profiles of people that have joined the program, and you'll see some of the topics and data sets that we will be utilizing in different sessions. Right here, you'll find the courses. So online sessions are going to be intermixed with some of the courses that you can take at your own pace, asynchronously. So you'll be able to complete these courses and benefit from some of those basic things so that the sessions that we have are more interactive and you can spend the time during the sessions practicing some of the skills that you've learned and explore different data sets and projects that we've prepared for the program. So as you go through the courses, you'll start seeing your progress and you'll be able to complete those and get an individual certificate of completion for each one of these courses. Some of the courses will have just theoretical background and some of them will have some practical assignments that you can complete. There are also going to be some projects that we will keep adding throughout the program so that those of you that have completed some of the courses can also try to apply the learned skills on some of these project data sets. Here we will also measure the progress. We'll look at how all of the participants of the program are making their way through the course material, practicing some of the pipelines that we will be discussing in the sessions, and we'll see how we can all achieve a higher level of interactive discussions so that we can start working on independent projects. So the basic idea is that we will keep track of your activity. You'll be able to see track of your activity, and we'll try to address any types of issues that come up as a result of this interaction. So if you have not done so yet, you can also join this program today by completing your registration and start filling out your profile. So for all of the members that have joined, I do want to ask that you actually go through your profile, make sure that you have a full name, have some kind of a profile image or an avatar, and that way that information will be used in the program certificate as well as the individual certificates of courses that you will receive after completing the assignments and the quizzes including in that course. Another resource is the T-Bioinfo platform. The T-Bioinfo platform is really a springboard for you to launch your experience in bioinformatics. It offers a variety of resources that include some cloud-based tools that you can use to process large data sets. It gives you some guidance on how these tools work together by organizing them into logical pipelines, where each step of the way is explained with a detailed explanation of that method, as well as some links to where you can find the algorithms and potentially even the source code for some of the open source methods. And we'll also utilize some additional tools. Those additional tools will help you improve your ability to visualize data, interact with this data, and get some biological interpretation of the analysis results. Before I continue, let's see if there's any questions. So I'm just going to pull up in the chat. For those of you that have not yet received your login information, we will still be emailing you the access for the T-Bioinfo platform. And make sure that you understand that the difference between the educational portal and the practical portal is going to be based on a specific password emailed to your email. So if you could make sure that you add marketing@pine.bio to your contact list, that will help you receive any kind of information that will be needed for the program. Okay, so if you have any questions, you can type them right here in chat, or you can email us later and even post your questions on the forum. As I mentioned, the forum is the best place for you to post any kinds of technical issues or any kinds of questions that you might have. So let's go on to today's topic, bioinformatics and translational oncology research. So we'll talk briefly about cancer biology, looking at changes in organs, tissues, and cells, and thinking about some of the molecular factors in tumor development and metastasis, clinical and molecular data in oncology studies, and how we can analyze phenotype-genotype relationships. Okay, briefly, I'll answer the question that came up from MC. So in the platform is specified the pathway and also explain each step and which algorithms can be used. I will do a brief demonstration for those of you that have not had the chance to use the platform. So this is the T-Bioinfo platform. You can go on this link, server.t-bio.info. It's a cloud-based platform that includes many different tools for analysis of next-generation sequencing data, mass spectroscopy data, structural biology data, and provide some tools for data integration and modeling. You'll find that each section is organized by the data type and has a number of tools that you can use to process, integrate, and annotate some of the data. We have a set of demo pipelines that I'll show you. They don't include the steps for uploading the data, but you'll get a sense of what the platform includes. So here's the demo for RNA-seq. You'll find here that each step of the way explains the logic behind what we are trying to do. So as we start the pipeline, it will explain RNA-seq, how we will pre-process, map, and quantify levels of gene expression. You'll then see some options highlighted to limit based on what you are going to do in this pipeline. For example, we can use some pre-processing steps to eliminate some of the annotation, some of the technical variation. You can read about what this step does. You can also then connect this to mapping. For example, when we map using HiSat, you can read about HiSat2 and go to the article that describes what HiSat2 does. Then we need to construct isoforms and update the GTF file. And then we can map the reads to quantify the expression levels and prepare a table of expression. So the table of expression will look like this. And actually, by completing this pipeline, you will receive the downloads that a typical pipeline like that will produce. So by leveraging the platform, this is an example of an educational use. We can just go through the steps and explain them, see the inputs and the outputs, understand the role of each algorithm in a pipeline. But in the program itself, we'll actually use the live version of the platform. For those that have completed their registration, you will be able to use this on specific curated data sets that we've prepared for this program. But you'll also be able to apply this data, apply this platform or this analysis that you've practiced on to new types of data sets that you prepare as your own independent projects towards the end of the program. So the objective of the program will be to provide you with some basic tools like these that you can quickly launch your analysis. And once you've launched your analysis, you can take it as far as you want to with the tools that we provide or with some of the other tools that we will cover, as well as some of the open source tools like RStudio and others. Okay, so hopefully MC, that answers your question. Yes, I will happily explain about projects, but let me go first through the topic of today's session, because the projects will start making a lot more sense after we review some of the basic concepts that we are going to introduce in today's session. So bioinformatics and translational oncology research. Let's talk briefly about the molecular components, the biology of cancer. And then we'll talk about some of the data types that allow us to study this biology at the tumor and the tumor microenvironment levels. So what happens to a cell to make it become cancerous? 30 years ago, scientists could not offer a coherent answer to this question. They knew that cancer arose from cells that began to proliferate uncontrollably within the body, and they knew that chemicals, radiation, and viruses could trigger this change. But exactly how it happened was a mystery. Research across the last 3 decades, however, has revolutionized our understanding of cancer. In large part, this success was made possible by the development and application of the techniques of molecular biology, techniques that enabled researchers to probe and describe features of individual cells in ways unimaginable a century ago. Today, we know that cancer is a disease of molecules and genes, and we even know many of the molecules and genes involved. In fact, our increasing understanding of these genes is making possible the development of exciting new strategies for avoiding, forestalling, and even correcting the changes that lead to cancer. Cancer cells differ from normal cells in many ways that allow them to grow out of control and become invasive to other tissues. One important difference is that cancer cells are less specialized than normal cells. That is, whereas normal cells mature into very distinct cell types with specific functions, cancer cells do not. This is one reason that unlike normal cells, cancer cells continue to divide without stopping. Cancer cells have large, variably shaped nuclei. They are composed of many dividing cells with disorganized arrangements. Cells are of variable sizes and shape, and overall display a loss of normal features. So you can see here a comparison of normal and cancer cells. What we're looking at is called phenotype. So we can describe the different shape patterns and different properties or characteristics of cells and tissues to make a distinction from what we know of normal cells. In addition, cancer cells are able to ignore signals that normally cells pass to each other to stop dividing, or that begin a process known as programmed cell death or apoptosis, which the body uses to get rid of unneeded cells. Cancer cells may be able to influence normal cells, molecules, and blood vessels that surround and feed a tumor, an area known as the microenvironment. For instance, cancer cells can induce nearby normal cells to form blood vessels that supply tumors with oxygen and nutrients which they need to grow. These blood vessels also remove waste product from tumors. So the cell itself changes, but also starts changing the environment around it. And as the tumor grows, it actually sucks out a lot of the resources needed for normal cells around it. Around the tumor, the microenvironment actually includes multiple cells, including the immune cells. And cancer cells are also often able to evade the immune system, a network of organs, tissues, and specialized cells that protects the body from infection and other conditions. Although the immune system normally removes damaged or abnormal cells from the body, some cancer cells are able to hide from the immune system and evaded from being detected. Tumors can also use the immune system to stay alive and grow. For example, with the help of certain immune system cells that normally prevent a runaway immune escape response, cancer cells can actually keep the immune system from killing cancer cells. And so we can differentiate between hot and cold tumors. Tumors described as hot are those that show signs of inflammation, meaning the tumor has already been infiltrated by T cells rushing to fight the cancerous cells. The tumor cells have undergone many mutations and create neoantigens recognized by the T cells. The main idea behind checkpoint inhibition, for example, is to use antibodies to mobilize the T cell response. During the attack on the tumor, T cells become exhausted and lose important functions needed to kill tumor cells. The exhaustion is brought on by constant exposure to tumor antigens and signaling through checkpoint receptors. The antibodies block signaling through these receptors to prevent this loss of function. So what are cold tumors? In contrast, non-immunogenic cold tumors have not yet been infiltrated with T cells, a sign that the immune response is not working for one reason or another. The lack of T cells makes it difficult to provoke an immune response with immunotherapy drugs.
These types of tumors are mostly treated with traditional cancer therapies. Most breast cancers, ovarian cancer, prostate cancer, pancreatic cancer, and glioblastomas are typically cold tumors, which poses a problem for the widespread use of immunotherapies. The microenvironment surrounding cold tumors contains myeloid-derived suppressor cells and T regulatory cells, which are known to dampen the immune response and inhibit T cells trying to move into the tumor. So the tumor grows, recruits cells around it to form a specific type of a microenvironment, and that microenvironment actually can help us understand what types of mechanisms are being activated and how we could target the tumor growth that is going on. The main reason that cancer is so serious is its ability to spread in the body throughout many different tissues. Cancer cells can spread locally by moving into nearby normal tissue, and they can also spread regionally to nearby lymph nodes, tissues, and organs. And it can spread to distant parts of the body. When this happens, it is called metastatic cancer. For many types of cancer, it is also called stage 4 cancer. The process by which cancer cells spread to other parts of the body is called metastasis. When observed under a microscope and tested in other ways, metastatic cancer cells have features like that of the ordinary primary cancer, and not like the cells in the place where the cancer is found. This is how doctors can tell that it is a cancer that has spread from another part of the body. Metastatic cancer has the same name as the primary cancer. For example, breast cancer that spreads to lung cancer is called metastatic breast cancer, not lung cancer. It is treated as stage 4 breast cancer, not as lung cancer. So to remember these aspects of cancer biology, hallmark features of cancer were identified. These are distinctive and complementary capabilities that enable tumor growth and metastatic dissemination. These have evolved over the past decade and now include original 6, which is sustaining proliferative signaling, evading growth suppressors, activation, invasion, and metastasis, enabling replicative immortality, reducing angiogenesis energetics, and resisting cell deaths. An additional 4 have been added: deregulation, cellular energetics, avoiding immune destruction, tumor-promoting inflammation, genome instability, and mutation. So during this program, we'll talk about these hallmark features of cancer and how they can be studied using specific types of data. For example, we have to understand each cell to understand what type of cell is found in a specific tumor or the place where it is metastasizing to. So bioinformatics is a key aspect of characterizing the different cell types and tissues, as well as being able to understand what might be the reason for these cells to differentiate into cancer cells or even start traveling throughout the body and causing metastasis throughout different tissues. When we speak about bioinformatics, we're going to primarily speak not about the biology first, but we're first going to focus on the data. We'll talk about changes in the DNA, where we can identify specific genes and positions on the DNA and in the chromosomes, and then study mRNA transcription, so how that specific gene is translated into a functional mRNA and eventually a protein. As a result, we'll learn how to understand the processes that happen at the subcellular level and study specific data types and integrate them together to understand how this biology is reflected in the data. When we talk about translational oncology research, a lot of that type of research utilizes next generation sequencing. So in our next session, we'll talk about the process of next generation sequencing, and we'll see some examples of how this type of data could be processed. When we talk about the basic and translational oncology research, we'll talk about how these kinds of studies at different levels can inform anything from the basic understanding of the mechanisms behind cancer growth and metastasis, all the way to actual selection of treatments. And so we'll talk about different projects that will look at patient-derived cell lines, then patient-derived xenograft models, animal models that have tumors from people engraved into their bodies so that we can study the role of the microenvironment around them. And we'll talk about data from real patients where clinical trials of specific types of disease or specific treatments can help generate a collection of data that can help us understand how these basic processes that we can study in individual cells actually work in the human population. So how is this used by a doctor? How is it relevant for a clinician? A doctor can detect cancer once clinical signs are evident from available data, patient scans, x-rays, or blood tests. Once detected, a tumor is evaluated for malignancy. Criteria for malignancy include increased cell proliferation, loss of differentiation, infiltrative growth, and metastasis to other organs. In other words, we have to identify these hallmark features of cancer. If the tumor that has formed from these cells is still contained within tissue of origin, it is called in situ cancer. In situ cancer may remain contained indefinitely. If some cells experience additional mutations that allow the tumor to evade neighboring tissues and shed cells in the blood or lymph, the tumor is said to be malignant. The escaped cells may establish new tumors, metastases, at other locations of the body. The development of cancer cells is a process of deregulation of key molecular mechanisms. So monitoring those mechanisms along the way and seeing the impact of treatment on those mechanisms helps the doctor change the mode of treatment and identify potential new targets for a treatment to stop what's going on. A big part of that is what's called data mining. So data mining allows us to find patterns in data and then use those patterns for biological interpretation. But low throughput data like blood count, white blood count, heart rate, or even basic things like weight and size are simple to analyze. We can simply plot them visually and we'll find some patterns just like you see right here. So when we have low throughput data, for example, phenotypic data, that data helps describe what we see and provide us with a quick way to distinguish between certain conditions. For example, here you can see how size and weight can easily differentiate between men and women. But when we come to high throughput data, and this data has complex relationships that it describes, it really becomes difficult to just simply look at the data and understand what it means. So we'll talk about some of the methods of data mining and machine learning that can help us associate various profiles of individual genes, positions on the DNA, and their mutability to differentiate between these different conditions. But this basic mechanistic understanding of what's going on is even more complex when we talk about real patients. So we'll also talk about such studies as the Cancer Genome Atlas that allow us to take patient data and look at how real patients in different clinical trials grouped by different types of cancers and different types of other conditions can be studied to understand the need for integration of various molecular data types and association of those molecular data types with clinical phenotypes, with what is actually known about the phenotype of those patients. So now let's talk about some of the projects that we'll prepare. I'll just give you a couple of examples. One example is a project called Modeling Precision Treatment of Breast Cancer. This project, which was featured in Genome Biology in 2013, is interesting because the authors assembled a data set of about 50 different breast cancer cell lines that represent the whole spectrum of breast cancer variations. The cell lines have genomic, transcriptomic, and proteomic data, as well as an efficacy measurement of commonly used and experimental cancer drugs that include chemotherapy and different targeted treatments. This is a project that really helps understand how modern use of machine learning and big data can inform treatment selection in combination with high-throughput molecular data, data that can characterize different conditions at their subcellular level. We'll also talk about some of the clinical data that is available in projects like the TCGA. Liver cancer is an example where we will take a look at the different risk factors. For example, you can see here different types of risk factors associated with different hepatocellular carcinoma patients. And one of the factors, like we said, is hepatitis C and B that is prevalent in Asia and Africa. So we'll look at how clinical data that records ethnicity as well as viral hepatitis serology helps understand the risk and the causative agents behind liver cancer. So we'll actually take the publication that is called Comprehensive and Integrative Genomic Characterization of Hepatocellular Carcinoma, and we'll take at the data sets that they provide, including exome sequencing, copy number variation, mRNA-seq, microRNAs, and other data to help us understand the molecular profiles of hepatocellular carcinoma and the role of pathogens like hepatitis C and B in developing this kind of a condition. Do you have any questions about what we just reviewed? And Shipra, does that make sense about the project, or did you have any additional questions about the project? Okay, excellent. So does anyone else have any questions? Please again, post them in the chat. And what I would like to do next is review the program schedule and talk a little bit more in detail about future sessions that we will have. Okay, if there's no questions for now, I'll continue. So in our next session, we'll start talking about next generation sequencing and how we can use NGS data to analyze DNA variation. Okay, there's a question. What is a good way to retrieve papers with sequence data? Excellent question. We'll actually talk about the process of finding a good data set and evaluating the data set that can answer a specific question. So before you jump to looking for a specific data set, it's important to develop the project idea to a level where you can already plan the analysis steps and anticipate what kinds of results of analysis you will find so that you can look for the appropriate data to answer your question. So I think what we'll try to do is we'll learn about the different types of data. We'll then look at a few different examples and we'll see how that data is utilized in a number of different examples. And then we'll actually show you how to access some of these repositories and how to navigate, for example, NCBI, one of the largest collections of biotechnology information, to find sequencing data of different types produced by different mechanisms and representing different types of experimental designs. And then the question, this session will be posted on the forum. Yes, so definitely the recordings of the session, after we are done with the session, the recording will be posted on the forum and on our YouTube channel privately in the future sessions. And this session, which is the first session about the program, will be posted for everyone to see. So you will be welcome to see that as well. So in our next session, we'll talk about the different types of next generation sequencing. In the session after that, we will start talking about processing next generation sequencing data and the different steps that play a role in our ability to interpret the results of that processing. And we'll also talk about specifically gene expression or RNA-seq studies. Okay, another question here, can fatty liver stage develop into a liver cancer? Yes, we'll talk about different factors contributing to liver cancer, including how the process, it doesn't happen overnight, right? So there are stages, and some stages could be reversible. And then there are different genetic or pathogen related or even environmental factors leading to different types of cancer. So we'll talk about that in one of the sessions. After we study DNA variation and gene expression profiles, we'll start talking about machine learning for gene expression data. When we come to big data sets, for each sample, we have thousands of genes that could be characterized. One of the challenges is to find meaningful patterns associated with a specific phenotype. And so machine learning has the ability to help us to mine and explore the different patterns and use classification and feature selection to identify the links between the phenotype and very specific patterns that we've identified in genes or combinations of genes. We'll then, to answer your question, talk about different data sources to be able to find data to study oncology. And we'll talk about different types of data that is available. For example, we'll talk about SRA or sequence read archive data sets where you can find raw read data. So you can find FASTQ files and SRA files, and those will be used to first process and map all the different types of variation. As you know, variation is based on the type of analysis that you do to that data and the way that the data was prepared. So we'll talk about those different factors. We'll also talk about a repository called Gene Expression Omnibus, or GEO. That data set or that database contains a lot of different processed data sets. So data sets that already have gene expression tables. But it's also important to understand how those tables were prepared. Not all tables are going to be used for different types of downstream analyses. For example, for differential gene expression, you'd have to find a specific type of data and potentially even pre-process it using different normalization techniques. So we'll talk about those as well. And then we'll talk about such data sets like the Cancer Genome Atlas that, as I mentioned, contains multiple types of omics per individual sample, including, for example, blood as a control sample for each patient, which helps us eliminate the germline mutations and focus on somatic mutations associated with cancers. We'll also talk about a database called COSMIC, which is a database that accumulates annotation of different mutations in specific genes associated with conditions and treatment response. So after that, we'll talk about viral genomes in the host transcriptome. So we'll talk about the detection of different viruses. These could be viruses that cause cancer. These could be viruses that cause inflammation, and they could be just the cause of infectious diseases. But we'll talk about how are those detected. And so when we talk about finding a viral genome, we actually mean finding specific reads that give significant indication for either a whole genome or specific proteins of that virus. And some of the challenges associated with preparing the right kind of data to be able to detect those when we take samples from patients or from other hosts. We'll then talk about analysis that helps us characterize those different genomes. A lot of times genomes of viruses mutate much faster than larger genomes from hosts like humans. And so we'll talk about evolutionary studies and phylogenetic analysis. And we'll talk about how we can map the differences in the genomic sequence to understand relationships between viruses that have been studied and are known and novel ones that potentially might be found in a specific patient or a group of patients. And then we'll do an in-depth discussion of different types of workflows and pipelines that are possible to study different aspects of genomic variation. And we'll talk about how do we extract the most information to talk about the biological significance of the analysis results. So we'll actually talk about how we can use this type of analysis to draw biological conclusions. And we'll talk about some of the details of the outputs so that we can make reliable and solid interpretation of those results. We'll also talk about pandemics. A big part of what we observe in the current pandemic is that people with pre-existing conditions are affected in a much more significant way than healthy people. And so death rates and mortality rates in an infectious outbreak like this actually highlights some of the challenges that people with pre-existing conditions, including cancer, including autoimmune diseases, might have. And so we'll look at how this example of this novel coronavirus pandemic actually highlights some of the previous topics that we've discussed before that, associated with these chronic conditions. And to understand the biology of host-pathogen interaction, we'll look at different types of considerations. Some of these pathogens that we've already described have specific treatments. There are some resistance elements for those pathogens to evade those treatments. We'll talk about those. We'll also talk about the process of vaccine design. For example, human papillomavirus already has a vaccine, so we'll talk about herpes virus and other types of viruses that have been the subject of vaccine design discussions for many years, and what are some of the challenges and opportunities in that space. So to continue with this program, again, I invite all of you that have already registered to go and start the process of exploring the different resources associated with this program. This program will have sessions that we just described, and for each session, there's going to be an associated resource. So for today's session, the associated resource is Introduction to Bioinformatics. Perhaps many of you already know enough about bioinformatics not to complete the Introduction to Bioinformatics course, but I do want to encourage you to do that, because one of the first things that we want to achieve from that specific course is to learn about you. We don't have time to interact with each one of you one-on-one, but the first step of this course is to fill out a form about you. And we would love to learn about where you came from, what is your level of education, what about precision medicine interests you, and what kinds of objectives do you have for this course or for this program. So please make sure that even for the sake of going through that process of letting us know more about you, please complete this bioinformatics or Introduction to Bioinformatics course. It does highlight some of the big biomedical data challenges and opportunities that are changing research and healthcare as we know it. And so a great example, one of the people that has already joined the program did an excellent job at completing the profile. So I didn't let him know before the session, but we did want to highlight Ebot Tabi is a clinical laboratory and biomedical scientist at ACPHS. I'm not sure what that stands for, but I'll be looking forward to finding out. And former NIH fellow in biodefense and emerging infectious diseases. And he is an instructor. So I do want to encourage all of you to complete your profile as much as possible, and also explore some of the, and there he added in the chat, Albany College of Albany and Health Sciences. Okay, great. Thank you for letting us know. And I do encourage all of you to try and complete your profile as much as possible. We do have a number of resources to connect, and we feel like this program is not only a way to learn about these topics and practice the analysis. Eventually, as you'll discover in any research project, a research project is a collaboration between a team of people from many different backgrounds. So hopefully we can help put together teams of collaborators with different types of backgrounds and different levels of experience to help contribute to an effective project. Okay, and a correction here, it's Albany College of Pharmacy and Health Sciences in New York in USA. All right, great. And Ebot, we are happy to have you here, and all of you as well that are joining this program. So let me briefly describe some of the other resources that you will find. Let's go to the educational place here. So as I mentioned here, you will find your profile. Some of your other profiles also, I don't want to go over anybody else's. We do have some of the participants of our previous programs here, and so we're excited to have you here as well. Sarah has been with us in several programs from Florida. Cassidy, Barnes, and others I recognize as well. So a lot of the people participating in these programs, I imagine, have an interest in continuing to develop your skills and applying those skills to new projects, and really looking at how those projects could be more comprehensive, more impactful, more significant. So we will bring in a number of experts in bioinformatics to help address those projects, and that will happen towards the end of the program. Okay, so I do want to point to this link about the syllabus. The syllabus is going to include kind of a timeline of all the different sessions that we just described. And I encourage all of you to go through this timeline and see some of the associated courses, and we will continue to add more information on here. So if you have any questions about this syllabus, you can reach out to us, and we'll go through the syllabus again next time to make sure that everything is clear. Oh, sorry, that was a private message. Okay, what are these different courses? The courses are going to include different basic skills. The courses will have an overview, and then you'll be able to see the curriculum inside the course. After you complete the course, you'll be able to get a certificate for finishing that course. And as you go through the course, you'll see the course results here increase. So one of the things that we've noticed is that a lot of people actually do not have, don't see the finish course button. So if you've started some of these courses, or you will start some of these courses, make sure that you complete all of the assignments and you finish at the end at complete course. You also see that each course has a number of different elements there. For example, inside a course, you will have a control section on the top. So we know that some of you might see offline time that you might want to take these resources with you. So you'll be able to use some of these control points here on top. So what are those? First of all, you'll see that your profile is going to come with associated points. I'll talk about points here in a second. And you'll get notifications. So those notifications will help you see specific elements of interaction with other people on the platform, or your own activity to keep track of what's going on. The course will include a course outline, ability to search for specific course topics, navigating between the different sections of the course. You can magnify any of the images that seem too small or unclear. You can magnify them. You can download the PDF of that lesson and take them with you. And also for those of you that struggle with small size font, you can increase the font size and of course close the lesson if you want to go back to the main menu. We also have online materials that you can use to study this offline, and you will see specific materials like specific words highlighted and explained. When you hover over that word, if you see it underlined, you can see the different specific terms that might be important to understand clearly. Some of the courses have just basic material with quizzes. Others have assignments, and those are hands-on assignments that are actually reviewed by us. You'll see that this assignment, for example, will ask you to run a pipeline, and sometimes you will have the ability to do it interactively on the platform, and then at the end, submit your assignment results. So those assignment results will be reviewed by us, typically in between different sessions. If you have any questions about those assignments, you're always welcome to post your questions either within the assignment or on the forum. And we've received multiple feedback points from previous participants of our programs and courses. For example, Ricardo here from Peru says that this course was spectacular. I feel that I learned a lot about transcriptomics from its definition, studying to learning how to apply algorithms in a simplified and easy way on the platform. So really, I wanted to highlight here that these courses are appropriate for any level. We had people from high schools that took the time to review these courses and gave us positive feedback, as well as faculty that really enjoyed taking some of the combination of practical and theoretical knowledge that these courses provide. So what are some of the course topics that are included in these courses? We'll talk about a whole series of topics in RNA-seq on transcriptomic data analysis, including transcriptomics 1, 2, 3, and 4, where it's where the processing of data, the types of sequencing data, the basic result of the table of expression is explained. Then in transcriptomics 2, we talk about statistical analysis, analysis of different expression levels, differential gene expression, and some additional factor regression analysis that utilizes regression to identify relationships between experimental design and gene expression levels. Transcriptomics 3 that talks about big data, data mining, and machine learning. And transcriptomics 4 that talks about single cell transcriptomics. We also have a series of courses on next generation sequencing genomic data, like whole genome sequencing and whole exome sequencing, where you will learn about the very basics, like Introduction to Genomics will teach you about different types of mutations and variations found in the DNA. And then Genomics 1 and 2 will talk about single nucleotide mutations or polymorphisms in somatic and germline, as well as copy number variation and the statistical analysis of accumulation of mutations in different regions. We will also talk about the microbiome and the virome, and so an important resource to understand the different sequencing techniques and ability to study individual viruses as well as communities of microbiome composition will be explained in metagenomics 1 and 2. So in our next session, we'll start talking about next generation sequencing. How do we start analyzing DNA variation? So again, the objective before now and then is to try and complete Introduction to Bioinformatics, an online course and a synchronous course. And next time we meet, we'll talk about the specifics of processing, analysis, and interpretation of DNA variants found in next generation sequencing data. Do you have any questions? I'd be happy to answer any questions that you might have now. Okay, so you can take your time to add your questions to the chat. The objective of this program is to combine the theoretical and practical materials that we have on the platform to actually get a hands-on experience to process some of this data, get certificates of completion, or in our next session in the beginning, we'll talk about certificates of excellence, where you can actually stand out from the rest by working on an independent project if you choose to do so, and get some assistance from us on how to do that. To get started, we'll provide you with some examples of projects that you can go through and understand how the data was analyzed, where the data came from, access the same data, and then try to innovate there, or apply the same skills that you learn in those projects to a completely new data set that will actually help you to find. As I see that there's no questions, I think that's all we have for today. I'm really excited about starting this program together with you. I'll be looking forward to getting to know you by looking at your profiles, and hope that you can complete as many elements in those profiles as possible. We do have a LinkedIn group that you're welcome to join, where we go through some of the job opportunities, research opportunities, projects, and different topics that are important to precision medicine and machine learning. So make sure that, Deepsha, maybe you could post a link to our LinkedIn group, as well as a Facebook group. And
again, for all of you that joined. And we'll look forward to seeing you again in our next session.